Hello, ladies and gentlemen. My name is Zili from the Institut for Mikroelektronik Stuttgart. It's my pleasure to present our work at IEEE Sensors 2021. My co-authors are from the Institut for Mikroelektronik Stuttgart and the Institute for Nature and Medical Science of University Tübingen, Germany. The title of our paper is Front-end Electronics for Beta Cell Function Monitoring with an integrated FOPP detector. According to the statistics from the World Health Organization, diabetes is on the rise. In 2014, there are more than 400 million adults have got the disease. And yearly, there are several million people died directly or indirectly from diabetes. And diabetes can lead to complications in other parts of the body. For instance, the stroke, the heart attack, kidney failures, and so on. There are mainly three types of diabetes. Type 1, the body does not produce enough insulin. Type 2, body produces insulin but cannot use it well. And there's also the so-called gestational diabetes. It's a temporary condition for some pregnant women. So our research work is targeted at the type 2 diabetes. We would like to study the mechanism of the insulin generation with the glucose concentration on the cell level. So if we can understand the problem better, then we might be able to introduce some medical interventions to treat the disease. Insulin is generated in the beta cells that reside in the islets of Langerhans in the pancreas. So in this microscopic view, we show a single islet of Langerhans of a mouse, and the beta cells are inside this islet. For a healthy person, the blood glucose concentration can be directly sensed by the beta cells, and an instantaneous regulation of insulin will be perfectly matched to the changing glucose concentration. However, for people who suffer from type 2 diabetes, a major reason is due to the excessive food uptake over a prolonged time periods. Because the excessive food take and then the blood glucose concentration is always very high, and then it triggers the beta cells to produce more insulin and to have the increased activity. And then this results in the production of reactive oxygen species within the beta cells and leads to a kind of gradual decline in the number of available beta cells. So there is a direct relationship between the beta cells' electrical activity in the form of membrane oscillatory bursts and extracellular glucose concentration. And we can use the so-called microelectrode array to detect or to measure the electrical activity. And this photo shows that we place islets of Langerhans on top of an electrode, and the burst activity of the beta cells can be detected. To understand the beta cell electrical activity, we would like to introduce a parameter called FOPP. It's the fraction of plateau phase, which is an excellent biomarker. If we see that the electrical bursts, which has the quiet and then the active phase and quiet and active phase. And then the FOPP is just the percentage of the active phases over the time. So a high FOPP percentage just corresponding to a high glucose concentration. And from this picture, we can see that with glucose level of 10 millimolar per liter, we have, let's say, the FOPP smaller than the case when the glucose concentration is 15 millimolar per liter. And if the electrical activity of the beta cells in the form of the FOPP can be accurately monitored, and we can understand the phenomenon better, and we might introduce some medical interventions to treat the problem. And the beta cell activity, it's a very slow process because the glucose concentration in the blood cannot changing very fast. So normally the recording will take a long time and result in a very large data volume. Now let's take a more closer look of the beta cell activity. Here is a recording of the mouse beta cells change with the glucose concentration from 3 to 10 to 30 millimolar per liter. We can see that the bursts here have different frequencies, different burst widths. If we take a zoom-in view of a burst, there are a lot of high-frequency single spikes. So here we would like to give some decisive observations. These high-frequency spikes are actually not of interest for FOPP detection or calculation. However, the bursts are important as they hold the information of the percentage of time with the beta cell activity. So if the signal processing chain can be tailored in such a way that we can only preserve these low frequency burst informations and get rid of these high frequency spikes, and then the system will be highly simplified and the data volume can also be reduced highly. In a conventional FOPP detection method, all the oscillatory signals are recorded, including those high-frequency spikes. So the recorded data has a very large volume, and then to transmit and to process these data will cause very high power consumption. And in our work, we would like to introduce a front-end electronics to provide an integrated solution for FOPP detection. So at the front end, we will already get rid of those high-frequency spikes and preserve the information of the low-frequency bursts and calculate the FOPP on chip. So this will highly reduce the data volume and also result in a low-power system, and it will be useful for advanced in vitro and vivo beta cell monitoring. This diagram shows the front-end signal processing scheme. Two electrodes, a signal electrode and a reference electrode, are connected to a capacitively coupled front-end amplifier. The flat band gain of the amplifier is defined by the ratio of the input capacitor and the feedback capacitor, and the DC offset from the electrode side is rejected by the capacitor coupling. And then the signal at the output of the front-end amplifier is further amplified by a programmable gain amplifier to a sufficiently large amplitude. And afterwards, a dual rail envelope detector consists of diodes and capacitor banks are applied to capture the signal envelope. And this dual rail design allows us to tolerate the DC offsets from the previous stages. And then the simulation shows that with the envelope detection, the burst information is very well preserved, while the high frequency, the FOPP irrelevant signals are filtered out. And afterwards, our dual threshold comparator is applied to translate the envelope into digital signals. And then these digital signals can be further processed in a digital unit to calculate the FOPP. In our work, we have designed an ASIC which can interface up to 64 electrode channels in XFAP XH018 technology. It occupies an area of 3.5 by 2 square millimeters and on a supply voltage of 3.3 volts. So the FOPP detection signal processing scheme presented in the previous slide is implemented as one of the test functions in this ASIC. So we have tested the signal processing chain using a test signal generated from a keysight waveform generator via some microvolt signal generation circuit. And then we can see that we are able to firstly amplify the small signals to a sufficiently large signal amplitude with very good signal to noise ratio. And then the envelope detection functions of the circuit works nicely. And it proves the effectiveness of the circuit, first with the amplification and secondly with the envelope detection. We're able to detect it each burst. And then it was to mention that the capacitors in the envelope detection circuit has a very wide tuning range, so the decay time of the envelope can be tuned to fit various burst shapes during the real beta cell monitoring. And then the detected envelope is really a low frequency signal and has sufficiently large amplitude, and then these signals can be easily processed by a low speed, low power comparator. And the output of comparator is then a kind of digital signal which will be used for the FOPP calculation. Now let me conclude the talk. In this work, the design of front-end electronics for beta cell monitoring is presented. It is used for the study and treatment for type 2 diabetes. And the unique feature of the design is kind of integrated solution for FOPP detection. And then the proposed integrated FOPP detector circuit, adopting a kind of differential envelope detector structure and keep the design overhead to a minimum, and the only FOPP relevant information is reserved and processed, and it results in a highly reduced data volume. And this allows us to further apply this solution to more advanced in vitro and in vivo beta cell activity monitoring. And then the FOPP detection concept, as well as the effectiveness of the amplification circuits, are all successfully validated by the measurement results of an ASIC implemented in XFAP XH018 technology. Now this ends my talk. I would like to thank the sponsors of the project and the project partners, and thank you very much for your attention.